Good afternoon, ladies and gents. It's me, and we're going to talk about viruses. Make sure that you have your copy of the notes open on a separate tab so that you can take notes while I'm talking. All right, we're talking viruses today. Viruses are extremely simple in structure, and that's what makes them kind of amazing. Viruses are nothing more than a piece of DNA or RNA wrapped in a protein coat. There's no cell, there's no Golgi, there's no nucleus, none of that stuff. Just DNA or RNA wrapped in a protein coat. This is a picture of a kind of virus called a bacteriophage. Kind of cool looking. In the photographs on the side, you can see bacteriophages attacking a bacteria. All right, so exactly how big are viruses? I'd like you to pause this screencast for just a moment and to go to a website on Google called Cell Size and Scale. If you type that into Google, use the website to put this list of things, great term, in order from largest to smallest, or smallest to largest, I guess, from 1 to 9. So see if you can figure out just how big viruses are. Go to the Cell Size and Scale website. Pause this right now. Okay, and now you're back. I'm assuming you went and did that. Now let's go ahead and move on. Viruses contain DNA and RNA and proteins, and they reproduce. So are viruses alive? Well, scientists say actually no, even though they have all those things that make them seem like they're alive. Viruses don't have a nucleus, they don't have cytoplasm or organelles, they don't have a cell membrane. They cannot carry out any kind of cell functions, and they can only reproduce when an infected cell builds new viruses. So science says actually they don't fit the criteria for life. We could say that they fit the criteria for life if we change our definition. We'd need to make an entire kingdom of life just for viruses. We haven't done that at this time. All right, so how do viruses get cells to replicate them or to reproduce them? If they can't reproduce on their own, what exactly is happening here? Well, the process by which viruses reproduce is called the lytic cycle. And I want you to take a look at the diagram on the right and then follow along with the words on the left. In the first picture, you see the bacteriophage, that's the specific virus in this case, attacking a bacteria. So the blue rectangle is the bacteria, and the green on the inside is the bacteria's DNA. So the virus is attacking that bacteria. It attaches. The first thing it does then is it injects its DNA. So that pink is its genetic material. And all that genetic material says is how to build more viruses. So in step number 3, the host DNA is degraded. It starts to fall apart so that the bacteria no longer has its own DNA. The only DNA it has is virus DNA. In step number 4, the host cell is forced to put together new virus parts because the only DNA that's left is the virus's DNA, and the virus's DNA says, build more viruses. So the bacteria doesn't know any better, so it builds more viruses. Eventually, in step number 5, the viruses break out of the cell. It's so full of viruses that they burst out, and they can go on then to infect other cells. So it's a cycle, and it's called the lytic cycle. Now, it is possible, let's say one other thing. How exactly do viruses make us sick? Well, when viruses are bursting out of our cells, and that's a photograph of. The cell is in kind of the bottom, and then all those little dots are viruses pouring out. When viruses burst out of our cells, the cells themselves die, they're killed, and we can experience pain like a sore throat or other symptoms from those cells bursting. All right, now onto the lysogenic cycle. Sometimes the lytic cycle, which is this process of being infected by a virus, takes time. Sometimes there's a time delay, and we refer to that as the lysogenic cycle. The lysogenic cycle is when viruses remain inactive within our cells for days, months, or years. You may have heard, for example, that people can get shingles from the chickenpox virus. That's when the chickenpox virus has been sitting inside them dormant for years and years, and then suddenly it reappears, usually when we're older, usually 60 years, 50 years, or older. So what exactly is happening in the lysogenic cycle? I'm not going to go into real detail here, but basically the virus's DNA is inserted directly into the host's DNA, and the virus's DNA is copied with the host's DNA during replication. But at no time does the cell start building the virus. It just starts copying the virus's DNA. Every time that cell divides, it copies the DNA. More viral DNA is passed on to the daughter cells then during mitosis. And this remains dormant. The virus remains dormant. Its DNA just keeps getting copied from one cell to the next until some triggering event. And what that triggering event is sometimes is a mystery. Maybe a person experiences some kind of stress or exposure to ultraviolet light, heat, chemicals like certain medications. But lots of times when shingles, for example, will pop out of the lysogenic cycle, it's a mystery why it happens to some people and why it doesn't happen to others. So what I need you to know about the lysogenic cycle is that it's a time delay for the lytic cycle when something is going a little bit slower. All right, there are different types of viruses. There are DNA viruses that have just DNA inside them. Examples include smallpox, chickenpox, herpes, HPV, and the common cold. There are also RNA viruses. RNA viruses go straight, instead of having to make RNA, they go straight to the ribosomes and have their proteins made very quickly. Examples of RNA viruses are the flu, measles, rabies, and Ebola. Now, there is one more kind of virus called retroviruses. You may have heard of retroviruses. The standard sort of example of the retrovirus is HIV. The retroviruses have something called, they have RNA, but then they also have an enzyme that's called reverse transcriptase. We're going to learn more about reverse transcriptase. We're going to learn more about HIV in another set of notes. It's actually these notes, but I'm going to skip HIV right now, and we'll come back to that later. So I'm going to skip the HIV stuff for later, and now we're going to move on to treatment and prevention. How do you treat and prevent viral infections? Well, for the most part, there are no medicines to cure viruses. So once you come down with a virus, there's nothing we can do. There are experimental drugs like Tamiflu. Tamiflu is described as a prescription medication used to treat the flu in people 2 weeks of age or older who have had flu symptoms for no more than 2 days. The World Health Organization recently produced a study that said Tamiflu basically doesn't work very well. So Tamiflu is not a great, it's still experimental, it's not a great cure. Vaccines are the only thing that we have to prevent viral infections. So what is a vaccine exactly? Well, a vaccine is actually getting a shot of a weakened form of the virus. Sometimes it's the virus itself, but we've killed the virus. Sometimes it's a mutation. We've changed the virus in some way. But whatever the vaccine contains in it, the body learns to fight off the virus so that if it ever encounters the real or strong variety of the virus, it's prepared to fight it off. So that's what a vaccine is. We'll learn more about that a little bit later when we talk about the immune system. All right, the last thing that I wanted to talk about are prions. Prions aren't viruses, but they're related to viruses, so we're going to talk about them here. Prions are, we're just learning about these, so maybe next year I'll have to say something different. Right now, what we think prions are infectious particles made of protein. They're very, 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 very tiny, so they're hard for us to study directly. They're even smaller than viruses, so you can't really see them under a microscope, so it's difficult to know exactly what's causing some of these diseases when we can't see them. So they are presumed or believed to cause what are called transmissible encephalopathies. I love that word. Transmissible means it's contagious. Encephalopathy refers to the brain and the disease or pathology of the brain. So these are contagious brain diseases, including a disease you may have heard of called mad cow disease. All prions, at least all prions in mammals, seem to affect the structure of the brain. Many of them cause holes in the brain. They are untreatable and they are always fatal. And what is particularly scary about prions is that they can be transmitted through ingestion. In other words, you can eat infected tissues. So mad cow was such a scary thing because it had to do with beef. And animals that were infected, we were concerned, and it appears to have happened, that people who ate the infected beef then developed a related disease in humans. It's not called mad cow in humans, but they died from it. It was 100% fatal. So now anytime that there's a cow that has mad cow disease, it's a huge deal. All the cows in the area are killed. And they're treated in a special way and trying to prevent any kind of infection. At this time, the United States believes our beef is completely free of mad cow disease and there's nothing to be worried about. All right, that's it, you guys. Let me know if you have any questions, and I'll see you tomorrow. Have a great day.